Hello, I'm Haohong Zhang from Haohong University of Science and Technology. Today, my topic is MGM. It's a microbiome foundation model for microbiome analysis. As we all know, microbiome community present in diverse environments and have implications in medicine, biotechnology, and agriculture. And with sequencing technologies, there are a vast amount of microbiome data stored in public databases like Magnify. And several supervised learning methods have tried to integrate this data for microbiome analysis. However, these methods are easily affected by label bias or inaccurate annotations. So that to address this, we introduce MGM. It's a transformer-based language model pre-trained by unsupervised manners on extensive microbiome data. We firstly assembled a large data corpus called microcorpus 260K, which contains about 260,000 microbiome samples downloaded from Magnify. And the genera in the samples were normalized and ranked by their relative abundance, and then tokenized into discrete representation, so that we can consider a sample as a sentence and the genera in it as a word. The order is abundance from high to low. And our vocabulary include 10,000 genera, and the input lengths are 6 into 512. Our model is a GPT-2-like models, which have 8 transformer blocks and 8 attention heads in it. The model is pre-trained by causal language modeling, and it could be adapted to the downstream tasks by transfer learning. We first evaluate the language modeling performance on 1,000 samples in valid data sets. We provided the first N% tokens in sample and let the model to generate the rest of them. And we found that when only 20% general were provided, the similarity exceeds 0.9. And we wonder if MGM could capture the taxonomic difference during the language modeling, even if this information is not included in encoding process. We found that the embedding of bacteria and eukaryote formed into 2 distinct clusters, and there was an outlier cluster mainly consists of arthropoda, which is not considered as a main components in microbiome analysis. And for downstream tasks, we first fine-tuned MGM on microcorpus 260K for a classification task. This task is to predict the magnified biome of each sample. And we found MGM outperformed other methods, include MGM without pre-training, traditional machine learning methods, and the source tracking method. And our model also performed well on 10,000 newly included data. And the embedding of MGM is more representative than the abundance table. And in case 2, we fine-tuned MGM on 2 intercontinental IBD cohorts to address the regional limitation. We found the pre-trained MGM outperformed the unpre-trained model. And if we use the target regions data to fine-tune the model, it could perform better. And in case 3, we fine-tuned MGM on a longitudinal infant cohort. And MGM could accurately predict the development stage of infants from different delivery modes. And we extracted the attention weights for the genera and found that the 2 delivery modes has different attention map. For the example, the bifidobacterium, a common probiotics, received higher attentions in vaginal delivery, and the Haemophilus, a well-known human pathogen, received higher attentions in cesarean delivery mode. And the sample embedding also reflects the dynamic changes in the microbial communities during the development stage. The embedding is more like their mothers. And we compared the general with the high attentions and the keystone general identified by DKI and found they have a large overlap. And in case 4, we fine-tuned MGM on TCMA, a tumor microbiome dataset. MGM demonstrated high diagnosis accuracy, and the different cancer reveal distinct attention map. So we want to identify several potential treatment targets by simulate the deletion of a genus by removing it from the rank encoding. And we find the removal of general such like Agrigentibacterium resulting in significant effects across most tumor types. And there are also several genera with effects specific to certain cancer types, such as Isarcella in IAD. And for case 5, we performed in silico perturbations on a Crohn's disease dataset. In this dataset, we simulate the enrichment by moving the genus behind the BOS token and simulate the reductions by moving it ahead the US token and compare the similarity with the healthy control. And we found that MGM captures the microbiome alteration aligned with the original study and also identified several novel microbial alternations that is not reported in original study, but confirmed by other studies. And in conclusion, MGM is the first transformer-based foundation model pre-trained on large corpus of microbiome data, and it could capture the spatial and temporal dynamics in microbiome communities and served as a versatile toolkit for various downstream tasks. And in the future, we plan to extend the pre-trained dataset to include more diverse data. These are the informations for this study. Thank you for listening. Yes.
Right.
Yes.
So why did you not do the
mask language modeling?
Yeah.
Yes. We tried it from the very beginning, but the performance on downstream tasks is bad. So we chose to use causal language modeling. And in future, we plan to use this model to generate some newly microbiome data. So it could be a better choice.
Thank you. Other one. This model used for the design the personalized microbiome or probiotics for in the future plan. It is difficult.
Yes, it's difficult.
Yeah.